So we want to talk about the coronavirus testing, how exactly the coronavirus is tested and how we are going to check whether you get a coronavirus infection, that is SARS-CoV-2 infection or not. If you want to know about the techniques, we are going to talk about two different approaches to test for the coronavirus positive cases. One with the RT-PCR technique, which is the reverse transcriptase PCR technique, as well as the real-time PCR you can call it. Apart from that, we'll also talk about the blood test, the 10 to 15 minutes rapid blood test kits that are available now. So we're going to talk about the mechanism of both this testing of the kits. Cannot even see that with light microscope. It's difficult. So what we need to do is that generally this virus infects our upper nasal cavity. So what we need to take a nose swab, which is known as a nasopharyngeal or oropharyngeal swab, as in medical terms. They take this swab, cotton bud. Simply the cotton bud that we take for ear, but these cotton buds are big enough and with the flexible head. So use this cotton bud to take a swab from your nose, inside of the nasal cavity, not exactly outside of the nose, but inside of the nasal cavity. Just take a sample of that, and that sample is enough to find out whether the virus is present inside that sample or not, because this is filled with droplet and the virus particle. If the virus infects you, if the virus is in your body, then the sample must contain the virus, and that sample is enough to find out whether the virus is present inside that sample or not, because this is filled with droplet, and that sample is enough to find out whether the virus is present inside that sample or not, because this is filled with droplet. Rapid test kit process. Now remember, in both these cases we need to take the sample is that nasopharyngeal or oropharyngeal swab that we take, which must contain the virus if you're infected. But for a blood test purpose, we only take blood as a sample. So the sample in this case is a simple needle prick. Now first let's talk about the PCR based testing, the RT-PCR based testing method. Now what we use for this method, let me tell you one thing very clearly before, that neither of this test is going to give you a standalone result, because RT-PCR test is not exactly a diagnostic test kit. It is a process for research purpose. It's not for the diagnostic purpose, because RT-PCR test is not exactly a diagnostic test kit. It is a process for research purpose. It's not for the diagnostic purpose. Because RT-PCR test is not exactly a diagnostic test kit. It is a process for research purpose. It's not for the diagnostic purpose. But we're using this for diagnostic purpose because we don't have any other option right now. Along with this blood test kit, both of them, they are not 100% accurate. They are 98%, almost 98% accurate. So there is a significant chance that 2 out of 100 patients, they will show a false result. Okay, there is always a possibility, but 98 out of 100 people, they will always give you a positive, correct result. You zoom inside of the swab, we have this droplet. In the droplet we have the virus. Okay, so let me draw, these are the virus that is present there. Okay, let's say the virus is there. Now the virus contains genetic material. Every single person, every single living organism, they have the genetic material. The virus have their own genetic material. In this case, the genetic material of the virus is RNA. Positive single-stranded RNA is present as a genetic material. Now the thing is, what we do with reverse transcriptase PCR method is, in PCR, this method, polymerase chain reaction, is a method to amplify double-stranded DNA. When we have the double-stranded DNA, if you take one copy of the DNA, with the help of PCR, we can make thousands and lakhs of copies of the DNA. That's possible with this method. So it's simple amplification method, just like a Xerox process. So in this case, in order to Xerox the DNA or make multiple copies of the DNA, what we need to know the sequence of the DNA. You need to know what you are going to amplify. For this reason, we need to know the sequence of the RNA. Sequence means what the RNA is composed of, because the DNA, RNA, they are made up with nucleic acids. Okay, for example, in RNA it's ribonucleotides, for DNA it's deoxyribonucleotides. So for this virus, we need to know the sequence of that RNA, and that sequence we already knew that. We know that sequence, and China provided us that sequence. China provided us that sequence from before. So we know about that sequence, so we can design the method to find out specific region of the DNA. So let's assume, let me draw a hypothetical sequence here. This is, let's say, the RNA. RNA is single strand like this, and DNA is always double stranded like this, okay, like a ladder, but RNA is a single strand like this. So for this RNA, there are multiple sequences, you know, there are different structures, either with A, C. So because they are made up with four things, A, C, G, T for DNA, and for RNA, they are composed of A, U, G, C. So because they are made up with four things, A, C, G, T for DNA, and for RNA, they are composed of A, U, G, C. Okay, we can find out that particular target. Now different companies are developing the kit, the RT-PCR kit, to find out the different portion of the gene. Okay, so in different nations they are using different kits, different segment of the DNA, different segment of the RNA to be scanned, that to be amplified. So Germany is using a different kit, Japan is using a different kit, China using a different kit, India can use a different kit. So in different country based, so they are trying to find out specific regions of the sequence that must be present in the viral RNA. Okay, that's our thing. So what we want to do is that sequence we need to target, we need to amplify that sequence, we need to make multiple copies of that sequence. Now by definition the PCR will make multiple copy of the DNA, but not for the RNA. Now by definition the PCR will make multiple copy of the DNA, but not for the RNA. Now by definition the PCR will make multiple copy of the DNA, but not for the RNA. In this case, the very first thing is that we have this RNA. Let me draw the RNA with red color. This is the RNA. So the first thing we need to make is a DNA from the RNA. And generally RNA is prepared from DNA. This is a conventional method, a conventional process. RNA is always prepared from DNA. This process is known as transcription. Okay, but in this case DNA needs to be prepared from RNA, which is a reversal of the conventional process. So we call it reverse transcription. That's why the name came RT, reverse transcription PCR. So when we run this process again and again, generally PCR process is a cyclic process. We need to run it for several hours, 1, 2 hours, 1.5 or 2 hours. So when you continue to run this process, we are going to see that the moment they are making more and more double-stranded DNA, we are going to see more fluorescence. Okay, so as a result, what we can find out is that we can simply see a spike in the fluorescence like this, a fluorescence increase with time. With time, the fluorescence, this is fluorescence here, with time you can see an increment in fluorescence like that. So by seeing this fluorescence increment, we can find out, yes, the target DNA, the target region of the RNA, the viral RNA is being amplified. So this gives us a positive result, that if we find out that after this process or during the process of RT-PCR, if we find out the increase in the fluorescence, that means our target RNA region is being amplified. If our target RNA region is amplified, that means the person, that means the swab contained the viral RNA. That means the person is infected with the virus. So we back calculate and find out the tests to be positive. On the other hand, if you see there is no change in the fluorescence, if you see no change in the fluorescence after 1.5 hour, 2 hour or that time, we can say the test is negative. This is a positive test. Remember, this is how the test results are obtained. In contrast to that, we have a second test that is now being famous. In USA it's being used, in UK it's being used. Currently in India it's been launched and already all the kits are already been booked. So they are making a second batch. You need to pre-order the kit to test yourself for a COVID positive or negative at your home just within 10 to 15 minutes. But this test is a blood test. Okay, now what is the difference here? In the blood test it's quite easier. You don't need to understand a lot of these molecular techniques and details. Just like the pregnancy test kit, if you are familiar to see the pregnancy test kit, similar kind of kit, it has one point here known as a B point, it has one point here known as a S point, and then it has a C region here. First things first is that this is S means sample. This is where we put the sample. This B region is where we put the buffer. Buffer solution is a small solution that is provided with the kit, a small liquid solution provided with the kit. These are antibodies and C is control. Okay, now at what principle this test is working? Now this blood test works based on the idea that you have already been infected with the virus and your body is already producing antibody against the virus to protect yourself. So there is a fundamental difference between these two tests. The very first test with RT-PCR is going to find out whether you're infected with a virus, and it's going to give you a data about whether you are currently infected with a virus or not, whether you've been infected currently or not. Okay, but this blood test is not going to tell you whether you're currently infected with a virus or not. It's going to tell you an arbitrary value, that whether you have been infected with the virus earlier, or you are currently infected with the virus, or you are infected with the virus at some point of your time at the earlier stage. That's what this test is going to tell you. Because normally what happens when a virus, let's say this is the virus, infects us, after the infection our body's immune system is producing antibodies. Okay, the antibody will fight against this virus and try to kill the virus. So they are producing more and more antibody to kill this virus. So once the antibody is present in the body against a particular virus or bacteria, that is a proof that that virus already attacked you, because one antibody is specific against one virus, one antibody is specific against one type of bacteria. So this antibody means this particular virus. So the coronavirus antibody is a specific antibody, and we know what kind of antibody our body produces when a person is infected with a coronavirus, that is SARS-CoV-2 or SARS-CoV-1, either of that. This test kit, you cannot distinguish between SARS-CoV-1 or SARS-CoV-2, and we know what kind of antibody our body produces when a person is infected with a coronavirus, that is SARS-CoV-2 or SARS-CoV-1, either of that. This test kit, you cannot distinguish between SARS-CoV-1 or SARS-CoV-2. This test kit, you cannot distinguish between SARS-CoV-1 or SARS-CoV-2. This test kit, you cannot distinguish between SARS-CoV-1 or SARS-CoV-2. The similar thing, they will take that as a similar viruses, because the antibody type produced is similar. Okay. Now among these two types of antibody, body can produce both the type of antibody. Generally we call antibody as IG, immunoglobulins, IgM, IgG. IgM is known as the primary immune response antibody. So this is the antibody that our body normally produces at the first time when it is infected with the virus or bacteria. And IgG is the antibody which is produced for the second time when our bodies get infected with the same virus or bacteria. Okay, so if you have more IgG, that means you have been infected with this virus couple of times. When you have more IgM, that means no IgG, that means you've been infected with this virus for the first time in your life. And as I told you earlier in the prediction video, that we all at some point of our time been infected with this coronavirus, that we all at some point of our time been infected with this coronavirus, that we all at some point of our time been infected with this coronavirus, that we all at some point of our time been infected with this coronavirus, that we all at some point of our time been infected with this coronavirus. So it's nothing to worry about. So now let's see how this test will work. So it's nothing to worry about. So now let's see how this test will work. So it's nothing to worry about. So now let's see how this test will work.